Hello students, today we will discuss about the remaining cell organelles. Those are plastids. So what are these plastids actually? These are mainly present in case of plants and euglena and also some protists. Okay, these are present in the plants majorly. In some cases euglena also you will find these plastids and protists also. The term plastid given by the scientist name is Haeckel. Okay, so this plastid also considered as a semi-autonomous organelle. Why? Because these organelle also semi-autonomous. So these organelle also contain double stranded DNA as a genetic material, so able to synthesize some protein. So plastids are generally three types. These plastids are generally divided into three types by the scientist name is Schimper. According to Schimper, so plastids are generally three types. What are those? So leucoplast and chromoplast and chloroplast. Okay, leucoplast generally they are colorless plastids. So generally these example for the leucoplasts are amyloplast and alluroplast and elioplast. So leucoplasts are generally divided into three types. What are those? Amyloplast, alluroplast, elioplast. Leucoplast generally they are colorless plastids. Okay, these amyloplast generally stores the starch. Okay, so example for this is potato tuber. Okay, then alluroplast. These alluroplasts generally stores the protein substances, proteins. Okay, example for this is endosperm of castor seed and alluran layer of the maize. Okay, alluroplast generally stores the protein substances. So example for this alluroplasts are endosperm of castor seed and also alluran layer of the maize. Next coming to elioplast. So elioplast generally stores the lipid substances. So example for this is tuberose. So this is about the leucoplast. Next come into chromoplast. Name itself indicating chromoplasts are generally they appear in the red or pinkish color. Why? So because they contain carotenoids. So these chromoplasts generally contain carotenoids. So these are red or pinkish in color. So these are formed from the either leucoplasts or chloroplast. Okay, ఇవి క్లోరోప్లాస్ట్ నుండి కానీ అదేవిధంగా ల్యూకోప్లాస్ట్ నుంచి కానీ ఏర్పడడం జరుగుతుంది. సో దీస్ ఆర్ రెడ్ ఆర్ పింకిష్ కలర్. ఓకే, నెక్స్ట్ కమింగ్ టు chromoplast. These are the very important structures. Why? Because they involved in the photosynthesis. కిరణజన్య సమక్రియలో పాల్గొంటాయి కాబట్టి ఇవి మనకు చాలా ఇంపార్టెంట్. సో నెక్స్ట్ కమింగ్ టు chloroplast. So where this chloroplast is present? Generally in plants, green tissue contain these chloroplasts. So if you observe the chloroplast, so it appears in different shapes. Okay, so it appears in discoid shape in case of chlamydomonas. Okay, in chlamydomonas, so chloroplast appears in the discoid shape. అదే విధంగా మనకు స్టార్ షేప్ జిగ్నిమా సో నక్షత్రాకారంలో ఉండే క్లోరోప్లాస్ట్ మనకు సో జిగ్నిమా లోపల ఉండడం జరుగుతుంది. అదేవిధంగా గిడిల్ షేప్, ఈ గిడిల్ షేప్ క్లోరోప్లాస్ట్ వచ్చేసి మనకు యూలోథ్రిక్స్లో ఉండడం జరుగుతుంది. ఓకే, క్లియర్. సో chloroplast generally appears in different shapes. So first one is discoid shape present in case of chlamydomonas, second one is star shape present in case of zygnema. Okay, girdle shape is present in case of Ulothrix organum. Ulothrix is a alga. Okay, next coming to, so this is the, if you observe the internal structure of this chloroplast, so it is an ellipsoid shaped in general form. So it is an ellipsoid shaped structure, okay, or discoid shaped structure. They contain, okay, they contain outer envelope and inner envelope. Okay, outer envelope and inner envelope. So outer envelope generally so permeable to water molecules or some substances. So inner envelope forms a structures known as lamellae. Okay, inner envelope forms a structures known as so lamellae. So within the inner envelope you will find the matrix or stroma. So this matrix, this stroma, this region is called as stroma. Okay, so stroma is the site for the so dark reaction. Okay, so in the stroma, so particular structures are found. So flattened structures, so these are called as thylakoids. Okay, these structures are generally called as thylakoids. So these thylakoids, సో ఒక దొంతర్ల మాదిరిగా కైన్స్ ఒక విధంగా ఒక వరుసలు అమర్స్తే ఏ విధంగా కనిపిస్తాయో ఆ విధంగా కనిపిస్తాయి కాబట్టి వీటిని సో ఈ స్ట్రక్చర్ని మనం ఏమంటాం అంటే గ్రానా ఆర్ గ్రానా థైలాకాయిడ్. సో దీస్ గ్రానా థైలాకాయిడ్స్ ఆర్ ఇంటర్కనెక్టెడ్ విత్ ద హెల్ప్ ఆఫ్ స్ట్రోమా లామెల్లే. ఓకే, దీస్ గ్రానా థైలాకాయిడ్స్ ఆర్ ఇంటర్కనెక్టెడ్, ఒకదానికొకటి మనకు స్ట్రోమా లామెల్లే అనే స్ట్రక్చర్స్ ద్వారా కలపబడి ఉంటాయి. Okay, next in this you will find the lumen. Okay, in this grana generally you will find the lumen. So in the lumen generally during the photosynthesis, okay, during the photosynthesis photolysis of water will takes place. So lumen, generally in lumen the water molecule splits into so protons and as well as oxygen. So this grana is the site for the light reaction, where the in the light reaction generally so ATP and NADPH2 are produced, which is known as reducing power. So this is about the stroma, and in this stroma you will find the double circular DNA and DNA polymerase and also RNA polymerase, so and also ribosomes, 70S ribosomes are also present. Due to presence of these DNA and also ribosomes, they able to synthesize or manufacture some of the proteins. That's why these organelles are also known as semi-autonomous organelle. Okay, so generally because of presence of DNA polymerase and also DNA and 70S ribosomes, this chloroplast able to synthesize their own proteins, some of the proteins. That's why this known as semi-autonomous organelle. So now what are the functions of this chloroplast? Generally involved in the photosynthesis, and also which sometimes it also stores the fatty acid. So photolysis of water takes place and oxygen consumption, oxygen production also takes place, and also so CO2 consumption, whatever CO2 present in case of our environment, so that CO2 absorbed by the green plant, so which is fixed in the form of carbohydrates. These are the some of the major functions, and also involved in the assimilatory power production, which is helpful for the CO2 fixation. These are the some of the major functions of the chloroplast. With this the chloroplast structure is completed. Next we will discuss about the so single membrane organelles. So single membrane organelles, those are nothing but, so first one is lysosomes. So the lysosome discovered by the scientist name is Christian Didway. So these lysosomes are generally absent in case of mature RBC. So which is a single membrane bound organelle. Okay, this contain only single membrane. So this also contains the acid hydrolysis enzyme. Acid hydrolysis enzymes. Why we are calling it as acid hydrolysis enzymes? So acid hydrolysis enzyme అని ఎందుకు అంటున్నాం? So they works at between 4 to 5 pH. So these enzymes which are present in the lysosome, they act in the acidic pH. That's why they are knowing it as a acid hydrolysis. Example for the hydrolysis are what are those? So generally nucleases, cathepsin. Cathepsin is a protease, and phosphatase and lipase. So nearly 40 types of hydrolytic enzymes found in case of this lysosome. Okay, some of the example I have given, those are nucleases and cathepsin and phosphatases and lipases. So cathepsin is a protease. So these are the some of the acid hydrolysis. Now what are the functions of these lysosomes? So generally these lysosomes is also known as recycling units, okay, post-mortem units, and suicidal bags of the cells. So recycling units, why? Because so మన బాడీ లోపల ఏవైతే కణాంగాలు కానీ వేరే పార్ట్స్ ఏమైనా చనిపోయినప్పుడు సో వాటిని ఇవి విచ్చిత్తి చెందించి వాటిలో ఉన్న ప్రోటీన్స్ అదేవిధంగా అమైనో యాసిడ్స్ని మనకు ఏర్పరచడం జరుగుతుంది. ఈ అమైనో యాసిడ్స్ కానీ ఫ్యాటీ యాసిడ్స్ కానీ మనకు ప్రోటీన్స్ అదేవిధంగా లిపిడ్ సింథసిస్లో సో యూజెస్. దట్స్ వై దీస్ ఆర్ నోన్ యాజ్ రీసైక్లింగ్ యూనిట్స్ అండ్ పోస్ట్ మార్టం యూనిట్స్. సో మన బాడీ లోపల చనిపోయిన కణాంగాలతో సో కణాంగాలు ఉన్నప్పుడు ఈ లైసోజోమ్ విల్ గెట్ ఫ్యూజ్ విత్ దోస్ ఆర్గనెల్స్ and finally degrades. That's why post-mortem unit of the cell. So suicidal bags due to autolysis activity. Autolysis, what is autolysis? So killing themselves, cells are killing themselves. చంపవేయడం జరుగుతుంది కాబట్టి దీన్ని ఆటోలైసిస్ అని అంటారు, స్వయం విచ్చిత్తి కణాంగాలు అని అంటారు. This is about the lysosome. Now so based on the function and morphology, so lysosomes are generally so 3 types. Those are primary lysosomes, secondary lysosomes and tertiary lysosomes. Primary lysosomes, and secondary lysosomes, and tertiary lysosomes. Okay, based on function and morphology, so lysosomes are generally 3 types. What are those? Primary lysosomes and as well as secondary lysosomes and tertiary lysosomes. So what is primary lysosomes? Primary lysosomes generally contain hydrolytic enzymes, okay, which are involved in the hydrolysis of the so particular substances. And secondary lysosomes are nothing but they contain food material. So this contain secondary lysosome generally contain so food material plus lysosome together known as secondary lysosome. Next coming to tertiary lysosome. What are tertiary lysosome? Tertiary lysosomes are generally nothing but they contain undigested food. సో డైజెషన్ కాగా మిగిలిన ఫుడ్ పార్టికల్స్ని టెర్షరీ లైసోజోమ్స్ లోపల మనకు స్టోర్ చేయడం జరుగుతుంది. సో ఆల్టర్నేట్ నేమ్ ఫర్ ద సెకండరీ లైసోజోమ్ ఇస్ ఫాగోలైసోజోమ్ and heterophagosome and alternate name for this tertiary lysosome is residual body. So these lysosome also involved in the thyroxine formation in case of so animals. So this is about the lysosomes. Next we will discuss about the vacuole. So vacuole is surrounded by a single membrane known as tonoplast. Generally vacuole is a single membrane bound organelle which is covered by single membrane. That membrane is generally called as tonoplast. Okay. So what are the major functions of this vacuole? Vacuoles are generally present in case of plants only. They store sap, so food material and organic substances. Based on the function and storage capacity, these vacuoles are generally 4 types. What are those? Sap vacuole, contractile vacuole, so air vacuole and food vacuole. So based on the function and storage capacity, so these vacuoles are generally 4 types. What are those? Sap vacuole, contractile vacuole and air vacuole, and also food vacuole. Sap vacuole generally stores the secondary metabolites. What are the functions of the secondary metabolites? So secondary metabolites generally stores the, what are the secondary metabolites? Anthocyanin. What is the function of this anthocyanin? Which impart the floral organs or plant organs colors. సో ఈ యాంథోసైనిన్స్ అనేవి మనకు పుష్ప లోపల, ఫలాలకు, అదేవిధంగా సో మొక్క లోపల వివిధ రంగులు ఏర్పడడానికి కారణమైన వర్ణద్రవ్యం ఏంటంటే సో యాంథోసైనిన్, విచ్ ఈస్ ఎ వాటర్ సాల్యుబుల్ యాంథోస్ పిగ్మెంట్. ఓకే, నెక్స్ట్ వాట్ ఈస్ ద మేజర్ ఫంక్షన్ ఆఫ్ దిస్ sap vacuole, which attracts the pollinating agent during the pollination. Next coming to contractile vacuole. In case of amoeba and some of the organisms, this contractile vacuole involved in the osmotic regulation and also excretion. This contractile vacuole involved in the osmotic regulation and also excretion. Air vacuole. In case of some of the plants, you will observe the plants are floating on the water. How it is possible? Due to presence of this air vacuole. Okay, helpful in the buoyancy. This air vacuole generally involved in the buoyancy. And food vacuole. This food vacuole generally stores the food material. So this is about the vacuole. Next coming to this ribosome discovered by the scientist name is Palade. These Palade discovered the ribosome general. That's why these ribosomes are all called also Palade particles. So they do not contain any membrane around it. It is naked. So they generally the ribonucleoprotein only in case of this ribosome they do not contain any envelope around this ribonucleoproteins. Okay, this is about the ribosomes. And next coming to subunits. So in case of this ribosome, they are made up of 2 subunits. One is large subunit and then small subunit. So ribosomes are generally 2 types. What are those? 70S ribosomes and 80S ribosome. So 70S ribosome generally present in case of prokaryotes. So 80S ribosomes present in case of eukaryotes. These 70S ribosomes are made up of 2 subunits. So one large subunit called as 50S ribosome subunit and also 30S ribosome subunit. So eukaryotic ribosome is nothing but 80S type of ribosome, which contain 60S subunit and also 40S subunit. So what is S? So S is nothing but Swedberg unit. So Swedberg unit is equals to 1 into 10 to the power of minus 13 seconds. So generally the size of the ribosomes determined with the help of sedimentation coefficient in the through the centrifugation manner. So S is nothing but Swedberg unit. Swedberg unit is equals to 1 into 10 to the power of minus 13 seconds. Okay, this is about the ribosome. What are the major functions of these ribosomes? Ribosomes involved in the protein synthesis during the translation process. Okay, so which contain peptidyl transferase. transferase enzyme. So main function of this ribosome is nothing but involved in this protein synthesis. So this is about the ribosomes. Next coming to where this cytoskeleton actually present. So this is present in case of protoplasm. So which is a filamentous proteinaceous structures. Cytoskeleton is a filamentous proteinaceous structure. Okay, these cytoskeletons are made up of generally filamentous proteinaceous structures. So this cytoskeleton generally contain three types of protein filament. Those are nothing but microtubules, microfilaments. Another one is intermediate filaments. So these microtubules generally contain or made up of tubulin protein. Okay, microfilaments are made up of myosin and tropomyosin proteins. These intermediate filaments are made up of keratin, dynein, kinesin. So likewise, these three parts, microtubule contain tubulin protein, microfilaments are made up of myosin and tropomyosin proteins, and intermediate filaments are made up of generally keratin and dynein and kinesin proteins. So what are the functions of this cytoskeleton? Cytoskeleton is also known as cytomusculature. So generally it is, which is involved in the motility of an organism and also maintains structure and also involved in the spindle fiber formation during the cell division, spindle fiber formation. So this is about the cytoskeleton. Okay, the major function of this is maintain structure and involved in the motility, so spindle fiber formation. This is about the cytoskeleton. Next coming to cilia and flagella. Cilia and flagella. సో ఇవి మనకు cilia అనేవి జనరల్గా యానిమల్స్లో ఉంటాయి. సో ఫ్లాజెల్లా అనేవి ప్లాంట్స్ లోపల మేజర్గా ఉండడం జరుగుతుంది. సో cilia, these flagella are similar in structure. They are similar but they are showing some of the differences. What are the similar differences? So cilia, first we will discuss about the cilia and flagella differences. Okay, cilia is present generally 100 to 1000 in number in a cell, but whereas flagella, if you take flagella, only 2 to 4 flagella present in a cell. Next coming to, they are small in size and they are large in size, and these cilia are formed కణం చుట్టూతా కూడా ఏర్పడడం జరుగుతుంది, but whereas this flagella form at one end only. Okay, these are some of the major differences between cilia and flagella. Internally the structure is similar, but the formation of the number and also size and also presence, so these are the major difference between cilia and flagella. Now we will discuss the internal structure of the flagella. Flagella generally contains centrally, okay, axoneme, okay, generally contain single doublets. This structure generally known as axoneme. So axoneme generally contain 2 singlets, and around this is surrounded by a plasma membrane. Okay, this plasma membrane generally, so inside the plasma membrane 9 doublets are present. Okay, these are the some of the, so 9 doublets are present toward the peripheral region. So this is the central sheath. so known as axoneme. So in this central sheath you will find the single doublet, single proteinaceous filaments, and here doublets are present, protein doublets are present. These protein doublets are connected to central sheath through the radial spokes. Okay, these peripheral doublets are connected to central sheath through the radial spokes. So how many radial spokes are there? So manaku 9 peripheral doublets unnai kabatti manaku 9 radial spokes will be formed, and these are also interconnected with the linker proteins. So peripheral doublets also connected with the peripheral proteins also connected with the linker proteins. So this arrangement is known as generally 9+2 arrangement. Why? Because peripheral 9 doublets are present, and centrally you will find the 2 singlets are there. That's why this arrangement is known as flagella. This arrangement is known as 9+2. So but whereas in case of prokaryotic flagella you will find the 9+0 arrangement. Why? Because central doublets are absent in case of prokaryotes. So this is about the structure of the flagella. The major function of the flagella is, they are involved in the motility and also some other uses with this flagella. This is about the structure of the flagella. Where these are emerged? They are emerged from the cytoplasm. So they produces current in the cytoplasm too, which is helpful for the movement of the cell. This is about the cilia and flagella. Next we will discuss about the centrosomes and centriole. What are these centrioles and centrosomes? They do not contain any cell membrane. Okay, they are naked. So centrosomes are generally present in case of animal cells only, main characteristic feature of the animal cell. Okay, so this centrosome is made up of 2 centrioles. So these 2 centrioles are arranged perpendicular to each other. Okay, centrosome is generally made up of 2 centrioles. So these 2 centrioles are arranged in perpendicular to each other. Around these, these centrioles are surrounded by a pericentriolar matrix. So ee centrioles chuttu kuda manaku pericentriolar matrix anedi undadam gumigudadam jarugutundi, pericentriolar matrix. So this pericentriolar matrix ఉండడం వల్ల మనకు ఇది ఒక కణాంగం లాగా కనిపించడం జరుగుతుంది. That's why this considered as a one cell organelles. Actually that is do not contain any membrane around these centrosome, but pericentriolar matrix is accumulated to surround around these centrioles. So what are the functions of these centrosomes? Before going to functions, we will discuss about the structure of the centrosome. So structure is similar to that of flagella. So here also this is surrounded, this is not surrounded by plasma membrane. So which is pericentriolar matrix is there. So peripheral 9 triplets, peripheral 9 proteins are fibrillar proteins are there. These are, so instead of doublets, so they are triplets in manner. So ivi 3 protein నిర్మాణాలను కలిగి ఉంటాయి. సో పెరిఫెరల్ 9 proteins are there. These 9 proteins are triploid, triple. సో 3 ప్రోటీన్లను కలిగి ఉండడం జరుగుతుంది. This is about the structure of the centrosome, and so centrally you will find the proteinaceous hub. This proteinaceous hub is present in the centrally. So these protein around this, the peripheral 9 triplet molecules are present to the peripheral region. So in this is accumulated with the pericentriolar matrix. So these 9 triplet molecules are attached to the central hub, proteinaceous hub, with the help of radial spokes. Here also these triplet proteins are interconnected with the linker proteins. So this is the structure of the centrosome. Okay, generally central core is made up of proteinaceous hub called as proteinaceous hub, which is surrounded by the 9 triplet molecules. So these 9 triplet molecules are attached to the central hub through the help of radial spokes. So these proteinaceous triplets are also interconnected with the proteinaceous fibers. Next, what is the function of these centrosomes or centrioles? Generally, produces spindle fibers. So what are the functions of these spindle fibers? These spindle fibers are involved in the chromosomal separation during the anaphase. So these will get stretch and contraction, and finally this separates the chromatids during the mitosis as well as meiosis. So the centrosomes or centrioles mainly present in case of animal cell, main characteristic feature of the animal cell. So this is about the centrosomes and centrioles. Next, coming to another cell organelles, that are microbodies. So micro name itself indicating microbodies. These are the cell organelles which are smaller in size. So means 0.5 to 1.5 micrometer. So length or diameter is there. So these microbodies described by the scientist name is Rodin. Okay, these microbodies are generally two types. What are those? So glyoxysomes and peroxisomes. Okay, first we will discuss about the peroxisomes. Where these peroxisomes are present? So in case of animal cells, it is present in the liver cells and as well as kidney cells. So what are the major functions of these microbodies? So involved in the respiration as well as oxidation. So peroxisomes are generally present in case of animal cells and plant cells. In animal cells, generally it is present in the liver cells and as well as kidney cells. So in case of plant cells, these plant cells, these are present in green tissue. Means photosynthetic green tissue contain the peroxisomes. Why? Because they produces during the photorespiration, they produces the H2O2 peroxide, which is toxic to the plants. So these are neutralized by the help of catalase enzyme, which is produced by the peroxisome. So these peroxisome generally involved in the detoxification and also neutralizes the methanol, other substances, methanol, formaldehyde, nitrates. So these are neutralized by the peroxisomes. So peroxisomes generally involved in case of plant photorespiration or C2 cycle, involved in the photorespiration. And these peroxisomes also contain D-amino acid oxidase, and also urate oxidase. Okay, this is about the peroxisomes. Peroxisomes are generally involved in the detoxification or neutralization process. In this, methanol and as well as formaldehyde and nitrate are degraded, and in case of plant cells, it is involved in the C2 cycle or photorespiration, where the catalase is produced and which is neutralized the H2O2 which is produced during the photorespiration. And these peroxisomes also contain D-amino acid oxidase and also urate oxidase. Next, coming to glyoxysome. So what are the major functions of this glyoxysome? So peroxisomes also, one more point. So here peroxisomes also involved in the glycolate pathway. So generally involved in the glycolate pathway. Next, coming to glyoxysomes. So glyoxysome generally present in case of plant cells only. So these are involved in the glyoxylate cycle. So in this process generally fatty acids are converted to carbohydrates. So they are involved in directly in the gluconeogenesis process. So they are mainly present in the, so in case of germinating seeds and as well as so fat rich cells. This is about the micro bodies, that is peroxisomes and so glyoxysomes. Next, cell organelle, that is the major cell organelle, that is nothing but nucleus, last cell organelle. So who discovered this nucleus? As I told you, so Robert Brown discovered the nucleus first. Okay. So this nucleus generally present in the eukaryotic cells. So what are the main characteristic features of this nucleus? So which is before going in details into this nucleus, so we have to know, so nucleus generally present, all the cells contain one nucleate, that cells are known as uninucleate cells. Example, so major all living organisms contain so single nucleate per cell. Some cells contain so two nucleates, those are, that condition is known as binucleate condition. So in these cells, each cell contain two nuclei. Example for this is paramecium. Okay. So and some of the cells contain so more than two nuclei. So that type of condition is multinucleate condition. So example for this is, so in case of animal cells, bone marrow is there. So bone marrow generally contain so multinucleate condition. So multinucleate condition in case of animal cells is known as syncytia. Syncytia is nothing but the condition, multinucleate condition generally known as syncytia in case of animal cells. Adevidanga plant cells. So plant cells, example for the plant cell, multinucleate condition in case of plant cell is so tapetal cells and as well as latex vessel. Okay, these tapetal cells and latex vessel contain generally so multinucleate condition. So multinucleate condition in plant cell so known as cenocytic condition. So multinucleate condition in case of animal cell is known as syncytia condition, but whereas so multinucleate condition in case of plant cell generally known as cenocytic condition. So next coming to structure of the nucleus. So if you observe the structure, so generally which contain two envelope. So as I told you, generally outer envelope is continued with the endoplasmic reticulum, isn't it? So this outer envelope generally continued with the so endoplasmic reticulum. So this is known as outer envelope. This is known as inner envelope. Okay. So the space between two outer and inner envelope is known as perinuclear space. So whereas in case of plastids, the space between two layers means outer and inner envelope is known as periplastidal space. In case of mitochondria, so there also you will find the outer and inner envelope. So what is the condition? So there you will call it as a perimitochondrial space. Perimitochondrial space is nothing but the space present between outer and inner envelope in mitochondria is known as perimitochondrial space. But whereas in case of nuclei, here also, so the nuclei contain outer and inner envelope. So in between the space is known as so perinuclear space. So in the within the nuclei you will find the so nucleoli or nucleus. This is called as nucleolus. This singular, this is plural word. So generally the inner content which is present in the nucleus divided into two. So contain generally two parts. What are those? Nuclei and another thing is chromatin. So now we will discuss about the chromatin structure. So chromatin, what are chromatin? So they are highly condensed, okay, they are highly condensed thread-like structures present in the nucleus. So these are generally, so in some stages during the cell division, so highly condensed forms the chromatin. These are chromatins are nothing but they produces chromosomes in case of the living organisms. So chromatin, if you observe the chromatin structure, you will find the 4 regions. What are those? So histone proteins, RNA, and as well as so non-histone proteins and okay DNA. So before going into details of the histone, RNA and non-histone and DNA particles, we will discuss about the chromosome structure. So generally chromosome is a rod-like structure which contains primary constriction. Okay, this region is known as primary constriction. So at this region you will find the kinetocores. So chromosome generally contain primary constriction, or primary constriction is also known as centromere. Sometimes at the end you will find also the secondary constrictions. So this secondary constriction contain part is known as SAT. Containing chromosomes are known as SAT chromosomes or satellite chromosomes, or these are also known as telomeres. Okay, so this is about the chromosome structure. Based on the centromere position, the chromosomes are generally divided into 4 types. What are those? So metacentric, next submetacentric, and acrocentric and telocentric. So during anaphase, so what will happen? The chromosomal separation will take place. So during the because of the stretching and contraction of the spindle fibers, so these chromosomes will move to the opposite poles. During this stage, so the chromomeres appears in different shape. So at this in case of metacentric chromosome, the chromosome 2 arms are, so chromomere or centromere is present in the center, so it appears in the V shape. But whereas in case of this submetacentric, so it appears as a L shaped, because so it is present near to the centromere. Okay, so in this case one arm is very long, one arm is very short. That's why this appears in the L shape. Whereas acrocentric, so it appears as a J shaped. So it is present, centromere is present at the near extreme end of the chromosome. So very small arm and very large arm will be produced in case of this acrocentric. So next telocentric, in case of this, so centromere present at the terminal region. Okay, based on this appears in the I shape. Based on the shape, chromosomes are classified into 4 categories. What are those? Metacentric, submetacentric, acrocentric and telocentric. So in case of metacentric, the centromere is present. So రెండు బావుల మధ్య లోపల ఉండడం జరుగుతుంది. So this appears in the V shape. But whereas in case of submetacentric, so it is present, centromere is present at the close to the end. It appears in the L shape. But whereas in case of J shape, so acrocentric appears as a J shape. So in this case, one chromatid is very small and another chromatid is very large chromatid. In telocentric chromatid, so that centromere is present at the end of the chromosome. So it appears as a I shape. Next, chromatin. So nucleum structures, all this we will discuss now. So chromatin. So what is chromatin? Which is a long thread-like structure which is highly condensed form. Okay. So which appears in the beads as a beads on string. These beads on string structures are known as nucleosomes. So if you observe the nucleosome structure, generally, so central core region is there. These central core region generally contain histone proteins. Histone proteins are acidic in nature. How many types of histone proteins are there? So 4 types. What are those? H2A, H2B, and H3 and H4. These are the 4 copies of, so 4 types of histone proteins. These are the basic histone proteins present in the core region of the nucleosome. So these are present in 2 copies, means each and every nucleosome core contain 2 copies of these histone proteins, means 2 copies of H2A, 2 copies of H2B, and 2 copies of H3 and 2 copies of H4. Totally 8 histone proteins are present. So these core is surrounded by a DNA or chromatin. Okay. So each core is surrounded by 2 turns of the chromosome. These 2 turns of the chromosome are centromere. Chromatin is 2 turns of the chromatin, generally around 200 base pairs length. So generally these nucleosome core contains surrounded by the 2 turns of the chromatin. This chromatin nearly 200 base pair length. Okay, this is surrounded by the 200 base pair length. So likewise, it is also surrounds another nuclear core. Okay, nuclear core. The DNA or chromatin present in between 2 nucleosomes are generally called as linker DNA. Linker DNA. So beads on string, nucleosome structures are found in the chromatin. So chromatins are nothing but, they are made up of central histone proteins which are surrounded by 2 turns of DNA. These 2 turns of DNA length is around 200 base pair. So between 2 nucleosomes the DNA region is present, that region is known as linker DNA. So what are the functions of the DNA nucleus? So which controls or regulates all the metabolism in case of living organisms. So with this the cell organelle topic is completed. Now we will discuss about the cell cycle. Till now we were discussed about the cell organelles and plasma membrane, all those things. So now we have to discuss about the cell cycle. So what is cell cycle? So cell cycle is nothing but duplication of genome and mother cell is converted to 2 daughter cells. So these events are represented in a cyclic manner. That cycle is known as cell cycle. So during what happens, during the cell division what will happen? కనైబజన్ జరిగేటప్పుడు మనకి ఏం జరుగుతుంది? సో మదర్ సెల్ నుంచి 2 డాటర్ సెల్ ఏర్పడడం జరుగుతుంది. అదేవిధంగా DNA which is genomic DNA will get duplicated. So these events are represented in a cyclic manner. This cycle is known as cell cycle. So cell cycle generally divided into 2 phases. What are those phases? So phase 1, that it is also called as interphase, and phase 2 it is also known as M phase or mitotic phase. Cell cycle generally divided into 2 phases. What are those? Interphase and M phase, or phase 1 and phase 2. So if you observe the cell cycle, so first you have the interphase. So interphase again divided into G1 S and G2, G1 and S and G2. So in this interphase generally, so whatever the energy or whatever the compounds required for the DNA duplication and cell divisions are synthesized in case of this interphase. That's why this interphase is also known as preparatory phase. Preparatory phase. Why we are calling it as a preparatory phase? So generally whatever the materials required for the DNA duplication and cell division will be synthesized in this phase. That's why it is also called as preparatory phase, and it is also called as energy phase. So why energy phase? So energy also required for the cell division. So this is in this interface, the energy whatever utilized for the required for the, so my M phase or DNA duplication or cell division, that is synthesized. So in case of this phase 1, that's why this is also known as energy phase. So interphase can be divided into 3 phases. What are those? G1 and GS and G2. So first G1. So G1 phase is also known as gap 1 phase. Okay. So if you take a cycle, so in that generally G1. So G1 is also known as post-mitotic phase. Why? One cell కణం కంప్లీట్గా మైటాసిస్ కంప్లీట్ అయిన తర్వాత, సో మైటాసిస్ కంప్లీట్ అయిన తర్వాత మళ్ళీ సో G1 ఫేజ్లోకి ఎంటర్ అవుతుంది కాబట్టి దీన్ని పోస్ట్ మైటాటిక్ ఫేజ్ అని అంటాము. సో హియర్ G1. సో వాట్ ఆర్ ద ఈవెంట్స్ టేకింగ్ ప్లేస్ ఇన్ కేస్ ఆఫ్ దిస్ G1? సో జనరల్లీ సో ప్రోటీన్ సింథసిస్ టేక్స్ ప్లేస్. Okay, then RNA synthesis and also ATP synthesis. So generally during G1 phase or gap 1 phase, the protein synthesis and RNA synthesis and ATP synthesis will take place. This energy is utilized in the S phase, because S phase actual duplication of the genome will takes place. So for this duplication of genome, the energy required, the proteins are required. Proteins are nothing but they are histone proteins. So next coming to another GS phase, that is called as S phase. So this S phase, so whenever the complete protein synthesis and RNA synthesis and ATP synthesis completed or perfectly produced these things, then the cell enters into S phase. Otherwise they go into the resting phase known as G0 phase. So synthesis of protein synthesis కానీ RNA synthesis కానీ ATP synthesis కానీ ఏదైనా సో S phase లో అవసరమయ్యే సో కాంపౌండ్స్ సింథసిస్ కానప్పుడు సెల్ అనేది రెస్టింగ్ ఫేజ్ లోకి వెళ్తుంది. ఆ ఫేజ్ నే మనం G0 phase అంటాం. సో నెక్స్ట్ కమింగ్ టు S phase. సో S phase is also known as synthetic phase. S phase also known as synthetic phase. Why synthetic phase? So here in this phase only the actual DNA duplication will take place. So if generally in S phase, okay, S phase, haploid genome is converted to diploid. Why? Because DNA duplication will be taking place and diploid cell be converted to tetraploid. Okay, actual DNA synthesis takes place in case of S phase, and here what will happen if the haploid cell enters into G1 phase, S phase, so haploid state is converted to diploid. If the diploid cell enters into this S phase, so diploid cell is converted to tetraploid. Why? Because the DNA getting duplicated here in case of this S phase. So next, another phase, if the DNA synthesis or DNA duplication is completed, then the cell enters into another phase is known as G2 phase. So G2 phase also known as pre-mitotic phase. Why? Because after this mitotic phase is there. That's why this phase is known as G2 phase. So what are the events taking place in case of this G2 phase? So in G2 phase, generally protein synthesis RNA synthesis, and also ATP synthesis, and also here cell organelles duplication also takes place. So what are the events taking place in case of this G2 phase? Protein synthesis and RNA synthesis and ATP synthesis and cell organelles. So as I told you, G1 phase also produces protein synthesis and RNA synthesis and ATP synthesis. So then what is the difference between whatever the proteins or RNA or ATPs proteins are synthesized in case of G1 phase? So these proteins are different from the G2 phase. So these proteins are nothing but they are histone proteins, because what are the proteins are required for the DNA synthesis that are produced in case of this G1 phase, but whereas G2 phase produced in G2 phase whatever the proteins are produced are synthesized that are required for the cell division. Okay, so remember those points. So next, if the protein synthesis, RNA synthesis and ATP synthesis and cell organelles get duplicated, then the cell enters into the another phase is known as mitotic phase or M phase.